So we'll continue with minerals, and before we do, just recap questions. What are some mineral analysis methods, or the principle or basis of some mineral analysis methods? What do we use to analyze minerals? Yes? AA is one. That's very good. Electrodes. Titration. Tara?
Colorimetric assays.
Colorimetric assays. You wanted to say something else, Erica? Were your hand up, or was Christina's hand up? Okay. So what are some considerations in sample preparation analysis? Yes, Michelle?
Contamination from the gasoline.
Contamination. So to avoid that, what do we do? Or to account for that, what do we do? We run a blank. You had your hand up, and now it's down. So volatilization during ashing, so you have to take care in the temperature of the dry ashing, Tara?
You always have to run a blank.
That's one of it, yeah. You want to say anything?
If you're trying to analyze iron, you don't grind it up in a metal.
Yeah, so we want to avoid metal contamination. Another thing you want to do when you're actually running the assay, you have to account for certain things. You want to make sure certain conditions are controlled, such as pH, temperature, and if you're preparing standards, you want to make sure you have kind of the same mixture or consistency. If not, if you're not able to have standards of the same consistency as your sample, what you would do, a very common method, is you do the standard addition method, where you spike your samples with standard, generate a standard curve within your samples themselves. This is something we talked about in spectroscopy section. Yeah, what are the reasons, some of the reasons for analyzing minerals? Jenny?
Nutrition labeling.
Nutrition labeling. Christina?
Water quality.
Water quality, so hardness of water, yes?
Toxicity.
Toxicity, we want to make sure we don't have high levels of toxic elements. Claire?
Health claims.
Health claims. There's one more, which is very important, Emily. Huh?
Shelf life.
What's that?
Shelf life.
Shelf life determination. For example, if you have some oxidation that will be prone to metal presence, then that would be important. Yonji, you wanted to say something else?
Fortified products.
Fortified products. So basically, if you do any processing that will result in loss of minerals, you want to make sure you account for that loss and fortify in the same amount that was originally present. Okay, good. What is a common method used to determine hardness of water? Yes, Erica? It's a titration method, and it's a common EDTA as a metal chelator. What does more titration measure? Yes, Amanda?
It measures the amount of chloride ions.
So it's a measure of salt in general by titrating the chloride specifically. Okay, and then how is it different than the Vollard titration? You have the more titration and Vollard titration. Both are titration methods, Bill?
Vollard is back titration.
Yes, and the other one is forward. The back titration meaning you add an excess of the reagent and then titrate the remaining reagent that was not used up by the chloride, whereas the more titration is just you titrate with the silver nitrate directly. Okay, very good. Remember, some of these questions will linger in exam. Okay, so we talked about AA in the AA chapter, and we talked about titration last time. Another common method for analysis of minerals is colorimetric determination. So basically, you react the mineral with a chromogen or a chromogen precursor to form a colored compound, and then you do spectrophotometry on it to determine, you run a standard, you apply Beer's law, and you calculate the concentration of your mineral that way. And this method requires ashing because you don't want any matrix interference. You don't want any turbidity from any matter in your sample. So everything is ashed and your minerals are solubilized the same way we prepare them for AA. A common method that is used quite a bit is for phosphorus determination using a molybdovanadate reagent. So it gives a very nice bright yellow color. This is a very common method, like I said. You generate standards and standard curve, and then you measure phosphorus in your sample. The ion-selective electrode works like a pH meter, basically, but your electrode is measuring ions, specifically certain ions. So we have a commonly used ion-selective electrodes for calcium, sodium, chloride, potassium, and fluoride. So the pH, the electrode, instead of it being sensitive to the presence of hydrogen ion, it would be sensitive to the presence of particular element ions. And then you measure the change in electric potential the same way you do for a pH meter. There was an equation in there, I deleted it, so you don't worry about it if you have it in your notes. Okay, so it is obviously responsive, the ion-selective electrodes are responsive to the activity of the ions. So you want to make sure if you are on standards to quantify, you will make sure your standards have similar ionic strength to your samples. Otherwise, you will have interferences and wrong results. And it's very sensitive. It can go down to 10 to -6 molar. But if you have that low of a concentration, it might take a while to get a reading. Same with the pH meter. You wait until the reading stabilizes. Some of the applications, the common ones, measuring salt and nitrate in processed meat, calcium in milk, salt and butter in cheese, and potassium and sodium in wine. So these are very common applications for ion-selective electrodes. Let's do some +2 exercise. Pull out a paper and let's do some fun calculations. Okay, read the question carefully. Consider the equation, the chemical equations up there in order to figure it out. Think of a strategy. You should be pros now in calculating moisture and ash and on dry bases. So this is just a refresher. I would like you at least to attempt to solve it. If you have an answer, let me know. So anybody on a lead? No? So, what's the moisture percent? Anybody?
80%.
80%. Okay, that's good. What's the ash on a dry basis? Yeah. Okay, so ash is 20%. Do we all know how to get these values? Anybody would like me to go through how we calculate these values? Okay, so how do we approach the last one, salt content of the original sample? What do we want, what's the systematic way of doing that? What's the first thing you want to do? Anybody wants to volunteer? Okay, Amanda.
You look at the KSCN added and then find out how much of the silver nitrate to reacted with,
Yeah,
to find out how much silver nitrate reacted with calcium, with chloride.
yes.
'Cause
So what do you calculate? Do you calculate mass or moles?
moles.
Moles. So you get the moles. Okay, so you're right there. So let's figure that one out. So first we said we added 30 milliliter of 0.1 molar silver nitrate. We want to know how much moles is this equivalent to. Okay, so 1.1 molar is basically 0.1 mole in 1 liter. So in 1,000 milliliter. What we want to find is how many moles in 30 milliliters. So basically 0.1 mole in 1,000 times 30 milliliter. What do you get? 0.0003? Yeah. 0.3 divided by 100.
2 zeros.
Just 2 zeros? Yeah, just 2 zeros. Okay. All right, so that's the original number of moles of the silver nitrate. So at equilibrium, number of moles of the thiocyanide would be equal to the number of moles of the remaining silver nitrate. It's 1-to-1 ratio. So 0.3 ml is, or not 0.3 ml, 0.1 mole again equal 0.1 mole in 1 liter. And then we have 3 milliliters, so 0.1 mole times 3 milliliters. So this is 0.0003 moles. Okay. So this is how much silver nitride remained after some of the silver reacted with the chloride. So this is the remaining. So the difference is the number of moles that were uptaken by the chloride. And since it's equimolar, 1-to-1, so the difference would be equivalent to the number of moles of chloride. Okay? So you would have 0.003 minus 0.0003 equal 0.0027 moles of Cl minus, which is equal 0.0027 mole of sodium chloride. Because you have 1 chloride and 1 sodium in 1 mole of salt. Okay? So now we know the number of moles of salt. I'll get the molecular weight, and then I can get the weight of the salt. So number of moles, remember, equal mass over molecular weight. I have number of moles, I calculate the molecular weight, which in this case would be 58.5. So 0.0027 equal the mass that I need to find over 58.5. So the mass in grams, this is grams per mole, this is in gram, this is moles, so that you know the units, and these will cancel, and then you'll get the mass in grams, which is equal to what? Can you do the math? 58.5 times 0.027? 0.158 grams. So the question is asking me salt content of the original sample expressed as weight by weight percent. So this is the weight of the salt. The original weight is 25 grams. So percent equal percent weight by weight of salt is equal 0.158 over 25 grams times 100. And that is, do you have that? So that's how you get it. Questions? Anybody would like me to repeat anything? Okay. All right, good. That's it for mineral analysis. Moving on to another great topic. Immunoassays. Very common analytical tool in food analysis. can you tell me what the principle of immunoassay is? Or have you heard of an immunoassay before? Do you know of an example of an immunoassay, Michelle? ELISA is an example. What does it stand for? Michelle? Yeah. Okay, so Michelle H. We have two Michelles. Okay. What was my question? What does ELISA stand for? This is short-term memory loss. Dory, you know Dory from, yeah?
I have part of it. I think it's like
Enzyme-linked immunosorbent assay. Very good. Okay, so what's the principle of it? Very briefly, yeah. Well, let Jessie go, yeah. Sorry, Simon.
We're going to do it like antibodies.
Yes, so we want antibody to interact with something we call antigens. So your antigen is what you usually would be analyzing in food analysis. So it would be either a protein antigen, could be for analyzing what? In what cases we would be analyzing proteins specifically using ELISA? Can you think of a case that's very important in food industry and labeling? Allergens. Yep. So, okay, so that is one. Other times we use ELISA for non-protein material. Can you give an example? Bacteria, pathogenic bacteria, using binding actually to the protein on the surface of the bacteria. Yep. What else? Yeah? Blood.
Like body fluid,
We use ELISA to measure blood.
like some body fluid, like water.
Not getting you, no. We use blood to get antibodies from the blood, yes. Okay, anything else you can think of? Toxins, pesticides, aflatoxin is very common procedure. GMOs, genetically modified organisms. So it is widely used in analysis, and there are several techniques that are considered immunoassays. ELISA is one, Western blot is another, lateral flow is another, which we will talk about in this lecture or next. Okay, so concept and theory. Just to refresh your memory, if you ever had this before, here are some principle. So it is based, like Jessie said, on antibody and antigen presence and interaction. So here's a typical illustration of an antibody. Antibodies are proteins. Again, see how important proteins are? They pop up in everything. Okay. So they have four molecules of proteins make up one antibody. So you have the heavy chain, one polymer, another polymer linked by disulfide linkages, and then you have the light chain, you have two of those that are also linked to the heavy chain by disulfide. So they kind of form a Y shape, and then here, between the one light chain and heavy chain, you have a place that is specific, highly specific to a region on your antigen, a very specific region. Often this region, not often, it is called an epitope. That region on the antigen that binds very specifically to the antibody is called an epitope. So there are a series of amino acids in the heavy and light chain that are very specific, and they have specific interaction and affinity to a region on the antigen. And they bind via non-covalent interactions. So it could be ionic, it could be hydrophobic, van der Waals, hydrogen bonding, but it's not covalent bonding. So how do these antibodies form in the body? They are often formed when the body is exposed to a foreign matter, or at least the body thinks it's a foreign matter. So B cells in the body, specific B cells, generate antibody, and some of these antibodies result in an allergic reaction. Not all antibodies result in an allergic reaction. So, for example, Ig, immunoglobulin E, is very specific antibody that will bind to a, let's say, protein and generate an allergic response. But what is used in food analysis is actually immunoglobulin G. So G are present in high amounts in the blood if they are exposed to a particular antigen. So these are the ones that are captured from blood samples to use in analysis, either captured from animal blood or they can be produced in vitro. So, like I said, the interaction of the antigen and antibody is very, very strong interaction, very specific interaction, and there are non-covalent interactions. And the affinity constants are very high because of the high affinity of the antigen to the antibody. So if our antigen is a protein, oftentimes you might hear a linear epitope versus a conformational epitope. So what does that mean? You know that a protein is made of it's a linear sequence of amino acids. But also it's not like a single strain. It kind of fold up and have a structure. So antibodies can interact directly with just a linear sequence, or it can have 1, 2, 3 amino acid in one area that come close to another 2 amino acids in a different area on the protein because of folding, and then it becomes 1, 2, 3, 4, 5, and that would become an epitope to an antibody. This is called conformational epitope. This is called linear epitope. So the size of the binding site is small, is not huge, and it's equivalent to 7 glucose units. And that's, I already said what an epitope is the region on the antigen that binds to antibody. Now, there is a polyclonal and monoclonal antibodies. You'll hear those terms quite a bit. So let me describe them a little bit for you. So, how do we generate antibodies? If we want antibodies for testing? What is the normal procedure? Huh? You inject an antigen into an animal. Very brutal, but you do that. So you inject an antigen into an animal, usually a rabbit or a mouse or a goat even. And then what happens, you collect the blood after a certain period of time, so the animal got exposed to a foreign body injected in the bloodstream. They will generate quite a bit of IgGs, antibodies. So you would get the blood, isolate the serum, and from there you can isolate the antibodies or use the serum directly in the assay. So that is basically a mixture of something we call polyclonal antibodies. What does that mean? You have so many different B cells in the body of the animal. Each B cell will produce an antibody with a specific binding site to a specific epitope. So, but there are millions of different epitopes that can bind to antibodies on the same antigen. So in the animal, you will generate so many different antibodies with so many different specificities to different epitopes. So that is called polyclonal. And this is very important in sandwich ELISA. You want to have polyclonal, and I'll explain that when I get there. Monoclonal antibodies, that means you have antibodies that are all the same. All of the antibody will react with one same epitope. That's what monoclonal is, and the way it's produced is actually because of a discovery where they were able to fuse B cells of a single B cell, single P cell basically, same characteristics, to cancerous cells. and then they do cell culture, and a lot of the same B cell is produced, and they all produce the same antibody with the same binding affinity to one epitope. So why is that important? It's very important in kits, ELISA kits, where you are looking for a standard antibody with one particular epitope selectivity. So there are applications for both, for polyclonal and monoclonal antibodies. So some characteristics of immunoassays. So basically your immunoassay, you want it to be specific, obviously, very specific to the antigen, no interference that you are looking to quantify. And oftentimes you want really high sensitivity. For example, for allergens, you want to detect allergens at really low levels for the sake of labeling purposes. So you want to make sure that your assay is very, very sensitive with a very low threshold. And also you want it to be simple. Everything you'd like to be simple. So we have, so as a must, we have to find the appropriate antibody for the antigen of interest that we are trying to quantify. And we want to figure out a way of separating bound antigen from non-bound antigen to the antibody in order to quantitate. And you want to make sure, in order to separate, you would use the capability of the hydrophobic binding of a protein to a hydrophobic surface. If you're able to do that, then you're able to separate bound from non-bound, which is the main basis of ELISA and Western blots. I already asked this question, how do we produce antibody specific? And that would be injected into the animal body. We already mentioned some applications, right? So what are they again? Repetition is always good, yeah? So allergens. We also said bacteria. We also said GMOs. We also said toxins. Oh, and there's another application I'll give you an example of at the end, is meat species authentication. Horse meat and beef patties.
Oh, geez.
So if you want to make sure you don't have horse meat, you will have antibodies for horse protein, and you can detect whether your patty has horse meat or is it just beef.
I remember hearing a story somewhere about there were, in the 50s, there were cases of people who would receive, I'm blanking on it, basically they were receiving immunizations, viral immunizations, but they were having a negative reaction to it because the immunizations, they were basically, the virus was injected into a horse, the serum was collected, and they were having a negative reaction because the people were actually consuming horse meat.
So no, I haven't heard about that, but if they are already pre-exposed to horse meat, then they would not have any the benefit of immunization. They will lose the benefit of the immunization, because immunization is you are giving the body small dose of something they are not used to having. So I guess something related to that.
Okay. It was, I just remembered that.
Yeah, I don't. Yeah, I haven't heard of that. Okay. So there are different types of immunoassays. You have the Enzyme-Linked Immunosorbent assay, which we will give examples about. Common ones, or the most used one, sandwich ELISA, comparative ELISA, and indirect ELISA, which sandwich ELISA can be direct ELISA and indirect ELISA. So we will talk about what does that mean in a minute. And then you have other immunoassays that are commonly used. Western blot is another one. It's a qualitative assay. Lateral flow is a field assay, so it's basically a strip test that you would use. We'll talk about that. And then immunoaffinity purifications, where you use affinity chromatography using antibodies and antigen principle to separate either antigens or purify specific immunoglobulins. Okay. So enzyme-linked immunosorbent assay, why is it called enzyme-linked? Because we use an enzyme that is bound either, we actually chemically bind it either to the antigen or to the antibody, and it would be our detection mechanism. So the enzyme that is bound, we will add the substrate in the reaction, and the substrate will convert to a product that is color, and that's how we detect, based on the activity of the enzyme and the conversion of a substrate to a product, and we relate that to the amount of antigen in the sample. So we'll talk about that in more detail. But the most common enzymes used are the horseradish peroxidase, very common, as well as alkaline phosphatase. Why? Because their activity is fast. They have fast activity. They are present in abundance. They're cheap and easily linked to the antibody or an antigen. And the production of a product is a simple mechanism using these enzymes. And with using enzymes and detection of an absorption of a product, we can use something called ELISA plate reader or microplate reader. And I'll show you a picture of that here. So basically, in this type of assay, you can run multiple samples, up to 96 samples in one shot. So basically, you add your reagents at a very small concentration. Each well can take up to 200 or 100 microliter, depending on the size of the plate. So you can add your reagents and plus a standard curve all in one plate, and then this would be a spectrophotometer, but instead of adding a place for a tube or a place for a cuvette, you will have a place to insert your whole microplate, basically, and it goes in, and absorbance in each well is read simultaneously. It's a very effective way of running an assay, where you can do tens of samples at one shot. You just have to be very good in pipetting, right? And you use oftentimes channel pipette. Who has seen channel pipettes before? So channel pipette is, you see what you use in the lab, it has one tip. You have channel pipette, which you have an arm, and you have a series of places where you can put tips, and then you can withdraw at the same time and dispense at the same time and on volume. It's very cool to have that capability when you're running a microplate. So what are the main steps in ELISA? This is a college board question, but also an exam question. So what you would need to do is, first thing, you have to bind the antigen or an antibody to a solid hydrophobic surface. Oftentimes it's a plastic surface, and the hydrophobicity of the plastic attracts the hydrophobic regions on a protein, and usually it's an antibody or an antigen if the antigen is a protein. So either you can start with an antibody solution or an antigen solution, and you allow it to interact, you allow it certain time, and then you do a washing step. You wash. There's also microplate washers that you can use. We have one in the lab that Chelsea uses for her immunoassays for allergens. So that's the first step. So you bind your first reagent, either antibody or antigen, and then you wash. There is a intermediate washing step. You wash out everything that did not interact. And then the next step, you want to block the surface, remaining surface. So, okay, I'm going to erase this and demonstrate how you would block the surface and why it's important. Okay. So you have a hydrophobic surface, and let's say in this case you are binding your antigen first. So you have your protein that binds via hydrophobic interactions, and you have other stuff in the sample that will just wash out. Okay? So you end up with a plate like that with the antigen. Okay, these we wash out. So now you want to add your antibodies, let's say. So you want your antibodies to interact with your protein. If you don't block these surfaces, what's going to happen is your antibody will react here and will react here. So, because your antibody is a protein as well. So you will have non-specific binding and you screw up your whole assay. So you don't want to do that. So before you add your antibody, before you do that, you don't do that. Before you do that, you want to block these surfaces. So you block it with an antigen that you don't care for and your antibodies are not specific for. Oftentimes in ELISA they use bovine serum albumin protein, or they use gelatin protein, very common to use, and block these sites so that your antibody only reacts via specific interaction to your antigen and will not interact non-specifically with anything else. Okay. So that's the importance of blocking. So after you put that first antigen or antibody, you block, you wash, and then you start adding the different components of the assay. If it's a sandwich ELISA, you start with an antibody, antigen, antibody, and then you put your substrate to the enzyme. And then at each step, you are washing, and then the final step is you add the substrate and you detect change in color after a certain period of time. Here's a sandwich ELISA. So as the name implies, sandwich ELISA is going to sandwich your antigen. So you will sandwich it between two antibodies. So the first thing you would do, you fix the antibody on a hydrophobic surface. The first thing is antibody is fixed. That will be the first layer. You block the open spots, and then you put your sample. Your sample will have antigen and other components. So you do a washing step and everything wash out, and what you have is your antibody and the antigen. Obviously there will be multiple antibodies and multiple antigens. And then because it's a sandwich ELISA, and here's where it's monoclonal antibodies come in handy. If you're running sandwich ELISA, you might start with serum, antibodies sourced from a serum. And then if you want to make sure that you label an antibody commercially, there's monoclonal labeled antibodies that you can purchase. So polyclonal here, monoclonal here would be really nice to have. So they will bind at a different epitope. Your antigen will have hundreds of epitopes on it if it's a large enough molecule. So the second antibody will bind to another epitope on your antigen, and it's labeled with an enzyme. And then you do a washing step for anything that did not get bound. And then the last step is you add the substrate, you generate the color and the product that is. And then you measure absorption. So we call them, they have specific names. So capture antibody, detection antibody. Okay, it's important to know these terminology. So this would be your antigen, target analyte, capture, detection, enzyme, substrate, product. And your antigen is sandwiched, that's why it is sandwich ELISA. Now, indirect ELISA, you can have a sandwich ELISA that is indirect. What is the direct versus indirect? Here things can get a little bit complicated. So you have the same principle. You have the antibodies on your plate. You have your target or antigen. Another antibody that is not labeled. Okay? And then you have an antibody to the antibody. Isn't that beautiful? Get an antibody to the antibody. So how do you do that? You basically, if you have a mouse antibody, you inject it in a goat. You get a goat antibody to the mouse antibody. And that antibody is linked to your enzyme, the horseradish peroxidase, and then you get the product. Now you're going to say, why do I need all this complication? Because these are easy to generate. To generate an antibody to an antibody is easier to generate and to label, so you can find them commercially. And then you can use actually the sera that has your antibodies, and you can use them to capture in both ends, to sandwich your antigen in both ends, and then you can get an antibody to this antibody and have it be labeled. So just from a commercial perspective, this would be a more common route to go. So ELISA, but indirect. When you hear the word indirect, you have an antibody to the antibody. When you don't have two antibodies, then it is direct. Okay, so with that, I'm going to stop here and continue with comparative ELISA next time on Friday. Try to review ELISA before you come Friday so that we can continue